Hello everybody. Today I'll talk about G protein coupled receptors and signal transduction. Now this is the first part of a series of 2 lectures, and in this lecture I have used several molecular dynamics animation, video clips as well as figures from different scientific publications, including my own research work in this field. For your convenience and further reading, I have referenced the source of the animations in these lecture slides, and also provided the list of publications and web pages at the end of the presentation. Now first I like to define what is signal transduction. Signal transduction or cell signaling is the transmission of physical or chemical signals or stimulus from a cell's exterior to its interior. The signals received by the cells must be transmitted effectively into the cell to ensure an appropriate response. I like to elaborate these points further. Now, the physical stimulus can be different types. For example, it can be a simple touch or it can be temperature. Now, you may want to know how we perceive this stimulus because of transduction of these signals or stimulus. Now, there are touch or pressure receptors as well as there are temperature receptors on the cell membrane, which are mostly ion channels. And these ion channels usually do not allow ions to move across the membrane. But under certain conformational shape, the ion channels opens and this allows the ions to pass through the membrane. And usually cells maintain a differential ionic concentration across the plasma membrane. So the ionic concentration outside the plasma membrane is different from that of the inner cytosolic concentration. So when touch or temperature stimulus occurs, these ion channels gets activated due to conformational change, and as a result the ions move across the membrane, and this causes a change in the membrane potential, which is then converted to an electrical stimulus and conveyed to different tissues or other organs. For example, touching some hot object can create an electrical stimulus which is conveyed to the muscle cells, which then contract and help us to exhibit a withdrawal reflex. Now, similarly, the chemical stimulus can be of different types. For example, it may be hormone molecules such as insulin or glucagon that can cause a change in the cellular glucose metabolism, or it may be nutrients. For example, the taste bud receptors in the tongue are sensitive to certain components of food, for example, sugar or acid, etc. Now, I also like to give an analogy to elaborate it further. For example, in this lower panel cartoons, now it shows that a mail carrier or postman delivering a letter in the letterbox in a multi-apartment building, and here in this example the mailman can be considered as a stimulus or ligand which interacts with the mailbox, which can be considered as a receptor on the cell membrane of the apartment building. Now this apartment building can be compared to a cell, and the individual apartments within that building are similar to different subcellular organelles within the cell. So when the letter or the message is delivered, it can be further conveyed to a specific apartment by some person living in that building, and in this analogy that person is activated protein or a second messenger, and the delivery of the messenger message to a specific apartment in that building may initiate further activity in that subcellular organelles. For example, it can bring happiness or celebration, thus initiating a physiological response. Now, there are different types of cell signal transduction pathways, and in this slide, I'll try to give you an overview of the major signal transduction pathways, and I'll highlight the differences. Then in the subsequent slides, I'll try to explain mainly the G protein coupled receptor mediated signal transduction. Now, transmission is mediated by a variety of biochemical changes within the cell, and which is also mediated by different receptors and their interacting proteins. And these receptors and their interacting proteins are present on biological membranes, for example, cell membrane, or it can also be present on the intracellular organelles membrane, for example, endoplasmic reticulum membrane or nuclear membrane or mitochondrial membrane. And the receptors that initiate these biochemical changes may do so by activating intracellular messenger molecules or by alteration in the cell membrane potential by the movement of ions in or out of the cell, or directly via intrinsic enzymatic activities within the receptor. So based on these properties, the signal transduction receptors are categorized into three major classes. Number one is receptors that are coupled inside the cell to G proteins, and these are called 7 transmembrane receptors. For example, muscarinic acetylcholine receptor is coupled to trimeric G proteins, and these are G alpha, G beta, and G gamma, which then lead to the generation of second messengers. The second group is called ligand-gated ion channels, for example, nicotinic acetylcholine receptor and serotonin receptor. And the third type is the receptors that penetrate the plasma membrane and have intrinsic enzymatic activity or are enzyme associated, and these are called enzyme-linked receptor, for example, insulin receptor, which has a kinase activity. Now, one way the nerve cells communicate with their neighbors is by sending a burst of small neurotransmitter molecules, for example, acetylcholine. And you may have noticed that both muscarinic acetylcholine receptor and nicotinic acetylcholine receptor, as the name implies, both of them can bind to acetylcholine, the neurotransmitter molecule. And these molecules diffuse to the neighboring cells and bind to special receptor proteins in the cell surface. In this case, it is either muscarinic receptor or nicotinic receptor. And these receptors then open. It undergoes a conformational change. And in case of muscarinic acetylcholine receptor, this conformational change causes recruitment of G proteins, and which then generate a variety of second messengers, which I'll discuss in the subsequent sections. But in case of nicotinic acetylcholine receptor, binding of acetylcholine causes a conformational change, and which then opens the ion channel, and that allows the ions to move across the membrane and creating an electrical impulse, which then translates into signaling cascades or a physiological response. And these processes are very fast because the small neurotransmitter, for example, such as acetylcholine or serotonin, can diffuse rapidly across the narrow space between the cells, and thereby the channels can open in milliseconds, or allowing the ions to flood inside the cell. And that is the fundamental basis of neurotransmission. So based on this discussion, the muscarinic acetylcholine receptor and nicotinic receptor are different, though both of them are activated by binding to acetylcholine. In case of muscarinic receptor, there is recruitment of G proteins, which then generate second messengers. But in case of nicotinic acetylcholine receptor, it's an ion channel, it's a ligand-gated ion channel, and binding of acetylcholine opens the ion channel and allows the ions to move across the membrane and generate electrical potential. The serotonin receptor is also a ligand-gated ion channel. Binding of serotonin brings a conformational change to the protein, which then allows the membranes, which then allows the ions to move across the membrane. Now, the receptor for insulin, which is a insulin receptor, is a large protein that is present on the membrane, and this protein passes the membrane, passes the message into the cell. The message is the presence of insulin in the external media. Now, it has several functional parts. There are 2 copies of the protein chains that come together on the outside of the cell to form a receptor site, and this receptor site binds to insulin, and this is connected through the membrane to tyrosine kinases, shown here at the bottom. When the insulin is not present, they are held in a constrained position, but when insulin binds, these constraints are released, and they first phosphorylate and activate each other, and then phosphorylate other proteins in the signaling network inside the cell. So they have kinase activity, which is different from the other ligand-gated ion channels of the G protein coupled receptors that I discussed. So at this point, I think we have some understanding of signal transduction. what are the different cell signal transduction pathways and what are their fundamental differences. So at this point, I'd like to discuss about the teaching learning objectives which I set for the remaining of this lecture. First, I'll discuss about the definition of GPCR and G proteins, and then I'll briefly mention a GPCR G protein cycle. Then I'll discuss the discovery and the basic structure of a GPCR and how GPCR structures and activation mechanism was deciphered. Then detailed crystallographic structure of the GPCR and the mechanism of activation following ligand binding. Then I'll talk about orthosteric and allosteric sites on the GPCR and orthosteric, allosteric, and bitopic ligand binding in the GPCRs. Then I'll discuss about the positive and negative allosteric modulation, the concepts of agonist and antagonist, and GPCR subtype specificity and ligand binding, and then I'll briefly mention the classification of GPCR, and I'll highlight why it is important to study GPCR and the importance of GPCR for therapeutic drug targeting. Now, what are G protein coupled receptors? GPCRs are the largest and the most diverse group of membrane receptors in the eukaryotes, and these cell surface protein receptors receive messages in the form of light energy, peptides, lipids, hormones, sugars, or proteins. A variety of different types of stimulus. For example, rhodopsin is a light-sensitive protein, is a light-sensitive G protein coupled receptors that is mainly found in the rod cells in the retina. The endothelin receptor is present on the cell membranes of vascular endothelial cell, and it binds to vasoconstricting peptides. Another example is a peptide hormone glucagon, which binds to glucagon receptor. There are also lipid binding free fatty acid receptors, which are G protein coupled receptors. There are hormone binding protein, GPCRs. For example, adrenaline binds to adrenaline receptor. And also there are proteins that bind to sugars. For example, the sweet taste in human is mediated by TAS1, R1, R2, or R3 GPCRs. Now, TAS1, R2, that stands for taste 1 receptor member 2. There are also proteins that binds to G protein coupled receptors. For example, addition G protein coupled receptor G1 or ADGR-G1 binds to transglutaminase 2, which is a protein, and control cell addition and cell-cell interactions. Now, what are G proteins? G proteins are named for their regulation by guanine nucleotides, GTP or guanine nucleotide triphosphate, or GDP, guanine nucleotide diphosphate. Some G proteins, such as the signaling protein RAS GTPases, are small proteins with a single subunit. Now, the RAS stands for rat sarcoma, reflecting how the first members of the RAS gene family was discovered. And the RAS family is composed of different human genes. They play a very prominent role in different human cancers. The G proteins that associate with the G protein coupled receptors are heterotrimeric. That means they have 3 different subunits, an alpha subunit, a beta subunit, and a gamma subunit. And in this figure, G protein coupled receptor is shown as a 7 transmembrane receptor, and there are G proteins which are shown as inactive protein complex, G alpha, G beta, and G gamma. And 2 of these subunits, alpha and gamma, are attached to the plasma membrane by lipid anchors. In the subsequent slides, I'll explain the significance of this attachment, how it helps the signal transduction through G protein coupled receptors. Now, at this point, as I discussed about signal transduction, defined G protein coupled receptor and G proteins, I'm now going to introduce you to a very basic G protein coupled signal transduction event, which is very fundamental and that lead to the production of cyclic AMP as second messengers inside the cell. And I'll use the example of a small hormone adrenaline, which is also known as epinephrine. Now, this is the messenger that tells a cell to ready themselves in danger and elicit a biological response, which is famously known as a fight or flight response. The adrenaline hormone is released into the blood from the adrenal glands, which are situated on top of the kidneys, and then it spreads throughout the body cells, where it is sensed by adrenergic receptor, which is present on the cell surface. So, when the adrenergic receptor is stimulated by adrenaline, the G proteins relay the signal on the inside surface of the cell membrane. And binding of adrenaline to its receptor changes the shape of the receptor, and it allows the receptor to bind inactive trimeric G protein inside the cell membrane. And this causes the G protein to expel the GTP molecule and replace it with GTP molecule. And the GTP causes a small loop, which is shown here in red color, to change its shape. And then the G protein falls into two pieces that frees the G alpha subunit with its bound GTP, then moves along the membrane as it is anchored to the membrane with a lipid molecule, and it eventually finds the enzyme adenylyl cyclase, which is also a membrane associated enzyme. And this little loop then binds to the enzyme and activates it. And the activated adenylyl cyclase then produces lots of cyclic AMP, which then spreads the signal throughout the cell. Now, eventually, the GDP in the active alpha subunit will break down into GDP, and the G protein will then reform in its inactive resting state, and it will be ready for the next cycle of signal transduction. Now, one major advantage of this approach is that it allows the signal to be amplified. Now, this is very important. A single adrenaline hormone molecule can produce a large amount of cyclic AMP due to sustained enzymatic activity of the adenylyl cyclase. So that is the fundamental basis of signal transduction. And then the cyclic AMP can then bind to different enzymes and activate the enzymes and can initiate a cascade of signaling events, which I'll discuss in the subsequent sections.